feel free to drop the questions on the chat box. We will answer your questions at the end of the session. And your respective feedback forms will be provided at the end of the session. So please fill the respective feedback forms by today evening. Now we can have an animated video of our college, Sri Vishnu College of Pharmacy. This is our campus, sir. Now I thank Principal Sir and Director Sir for giving us this opportunity for conducting this seminar. Now it's my privilege to introduce our today's guest speaker, Dr. Sanyog Jain. Dr. Sanyog Jain is working as a professor at the Center for Pharmaceutical Nanotechnology, Department of Pharmaceutics of Naipur, Mohali, India. Previously, he worked as a scientist at Defense Research and Development Organization, Ministry of Defense, Government of India. And he has he's an inventor of 16 patents, out of which 11 patents are already granted. And he has several prestigious awards, including Bharat Jyoti Award, Rising Suns in the Asia Award, Alkyl Amine Young Scientist Award. And he has secured a rank in the list of world's top 2% scientists. He has supervised 2 postdoctorates, 14 PhDs, and 105 master level projects. And he has patented several nanotechnology based drug delivery platforms and licensed tamoxifen sets technology for improved breast cancer therapy to industry for commercialization. Now, Dr. Jain is serving as an expert committee member of Indian Pharmacopoeia and member of Department of Atomic Energy, Government of India. Now the session, I may request the sir to continue the session with his presentation.
Yes. So a very good afternoon to everyone, and thank you organizers for a very nice introduction and a kind invitation. So let me share my slides.
Yes, sir.
So hope my slides are visible.
Yes, sir.
Yeah.
Well, sir.
Yeah. Okay, so I will talk on design of targeted nanomedicine for cancer therapy. Of course, this is just a small topic, but I will also discuss some other aspects of the drug delivery. Okay, it would not restricted only to the targeted nanomedicine. Okay, so my talk may continue beyond 1 hour also. So I hope you may be able to bear, because this is a last talk, and this is immediately after lunch, so students may feel sleepy. Okay, so now coming to the main topic. This chart shows various challenges in the site-specific or targeted drug delivery. So most of the drug, when we take in the conventional form, conventional drug delivery, they suffers from the uh, poor biodistribution and poor delivery at the non-specific biodistribution and poor delivery at the target site. Okay, so we take medicine either through oral route or injectable route. So irrespective of the route of administration, the drug reaches into the systemic circulation in the blood, and from blood it is rapidly eliminated by a urinary excretion or metabolism, and due to normal distribution along with the blood. Okay, so drug reaches at the non-specific tissues or organ, okay, where the drug action is not required, but the drug eventually cause side effects or toxicity. And there are several permeability related barriers, and due to which a very small amount of the drug it is able to reach at the target site. Okay, so to achieve the minimum effective concentration at the target site, so we need to increase the dose of the drug. Okay, so if we increase the dose of the drug, then a probability of drug reaching at the non-target site, it is also increased. Okay, so this is the problem. Okay, so can you imagine how much amount of the drug do we really require to treat any infection? Okay, so I can explain by a very simple example. Okay, suppose if a person is suffering from common cold and cough, okay, in which the microbial cell, okay, they colonize in the throat region only. Okay, and we take antibiotics, okay, to treat that throat infection. Okay, so any antibiotic you can name, okay, like ciprofloxacin, ofloxacin, azithromycin, erythromycin, okay, or any antibiotic. Okay, so what is the conventional dose, okay, of those antibiotic which we take? Generally 300 mg or 500 mg tablet, okay, they are available, and the patient is required to take uh, 2 tablets per day for 3 to 7 days, depending on the severity of the infection. Okay, so these antibiotic, okay, they kill the microbial cell which are colonized in the throat region. Okay, so how many total cells are there in our human body? Any answer? You might have read in the physiology, human physiology. Okay, so I will tell, there are total 10 to the power 13 cells in our human body. And if we assume, if one drug will kill one cell, one microbial cell, okay, then we will require 10 to the power 13 drug molecules to kill all the cells in our body. Okay, and if we want to convert this number into the mole quantity of the drug, so we need to divide by this Avogadro's number. Okay, so this comes around 10 to the power minus 10 moles. And if you want to convert this mole quantity of the drug into the gram or milligram quantity, then we need to multiply by the molecular weight of the drug. Okay, so molecular weight of most of the commonly used drug, it is less than 500. Okay, but let's, for the simplicity of the calculation, let's assume the molecular weight of the drug is 1000. Then still this quantity comes around 16 nanogram quantity. Okay, so theoretically this 16 nanogram quantity, it is sufficient to kill all the cells in our human body. Okay, but we are taking gram quantity of the drug. Okay, if I said ki the dose is 500 milligram and we are taking 2 tablets per day for 5 days, it means we are taking total 5 gram of that drug. Okay, 5 gram of the drug we are taking to kill few microbial cells. Okay, but theoretically the 16 nanogram quantity, it is sufficient to kill all the cells in our body. Okay, so what is the rationale? Okay, why we are taking so much or huge amount of the drug? Okay, so the simple answer is the needy cell, they are not able to take the drug is not able to reach at the target site. Okay, so this can be explained by this cartoon. There is enough food is available in the nest and with the mother, but kids, okay, these kids of this bird, they are still hungry and crying for the food. Similarly, there is enough drug is available in our systemic body pool, but needy cell, okay, they are not able to take the drug. Okay, so to deliver the drug at therapeutic concentration to the needy cell or to the target cell, we need to increase the dose of the drug. And if we increase the dose of the drug, then possibility of reaching the drug at the non-target site, it is further increased, and it also lead to development of the drug resistance and unnecessary side effects, and sometimes lead to treatment failure. Okay, so this is the major problem. And another sad story of the conventional formulation is the side effects and toxicity, which nobody wants. Okay, so what can be the possible solution? Okay, one possible solution is we can synthesize the new drug, okay, which can be more effective, more selective, free from any side effects or toxicity. Okay, but discovery of the new drug, it is very tedious job. It takes almost a period of 2 decades or 20 years. Okay, and out of the 2000 to 5000 lead molecule which are discovered in this early phase or discovery phase, hardly one or two molecules they are able to reach in the market. Okay, so there is no drug. If I say, okay, there is no drug is available for HIV treatment. Okay, what is the reason? Okay, the HIV virus changes its configuration very rapidly. Okay, so suppose if I develop a drug for HIV based on the present or today's configuration of the virus, okay, after 20 years, how many times the virus might have changed its configuration? Okay, forget about HIV. Recently, in just 2 years back, okay, since almost 2 years, the world is facing this COVID outbreak. Okay, so many drugs they have been proposed, and you are also aware that every time this virus is mutated and a new version of this virus it is coming, so the old therapy, okay, they are not effective. Okay, like when the first wave came, then it came that hydroxychloroquine, it is a blockbuster drug. Then in the second wave, remdesivir was the good drug. Okay, then third wave. Okay, so every time this different strains of the virus they are coming, and even it is efficacy of the vaccine, it is also questionable nowadays. Okay, because this mutated virus, okay, so it may escape from the earlier, it may not bind with the earlier approved drug or earlier developed drug. So these are the problem. It takes so many years. Okay, so by that time the therapeutic molecule lose its importance, therapeutic importance. Okay, this is the problem. And further, the cost of developing the new drug, it is increasing day by day, and in the next coming year, according to this survey, the cost will cross the roof. Okay, so that is the reason the majority of the pharmaceutical industries, they are now uh, uh, merging with the big pharmaceutical company like Ranbaxy. Okay, so it is now Sun Pharma. Okay, so many other big companies they have merged because they could not afford the R&D cost. Okay, so at this juncture I would like to state, in the old Darwinian economics, the big at the small, but in future the fast will eat the slow. So for India or any other country, okay, to succeed, we need to be smarter and faster. Okay, so here I would like to present the concept of drug delivery. So instead of synthesizing the new drug molecule, we can improve the delivery of the existing molecule itself. Okay, so by formulating in some suitable carrier system. Okay, so this approach, it is more cost effective, rapid. Okay, and it doesn't require any new drug discovery. It's just the formulation is changed. Okay, so commonly this approach is known as presentation of the old drug in new cloths. Okay, so this is the extension of my first slide. Okay, so when free drug is administered, it is rapidly cleared from the circulation. Okay, and it is inactivated by the like metabolic enzyme. It has higher affinity towards the non-target side. Okay, but it has poor affinity towards the target side. Okay, so the ultimate result is limited therapeutic effect and high toxicity. But when the same drug is formulated in some carrier system, so it is protected from premature degradation or elimination, and it limits the uptake by the non-target side, but it facilitates the transport of the drug towards the target site. Okay, so the ultimate result is high therapeutic effect and limited toxicity. So here this carrier system plays very important role in modulating or manipulating the normal biodistribution pattern of the drug. Okay, so these plays very important role in achieving the targeted drug delivery. So these carrier system, these are nothing but these are the nano carrier system. Okay, so they can be classified in variety of ways, but for simplicity I classified in like vesicles, okay, liposome and others, and particles. Okay, so they can be polymeric nanoparticle or lipidic nanoparticle and miscellaneous. Okay, so and this class includes dendrimer, nanoemulsions, micelles, etc. Okay, but again, if we think if we have prepared the drug loaded nano carrier system like a nanoparticle we have prepared, and when we inject them into the bloodstream, so we will be able to deliver the drug at the target site, then it's a big no. Okay, we need to consider and we need to understand what would be the biological fate of these carrier system. Okay, so what happens when these carrier systems, these are injected inside the blood, this nanoparticle. Okay, so they cannot leak out into the extravascular tissues like at the target site. Okay, they will confined into the bloodstream because these blood vessel, okay, so these endothelial cells, okay, which comprise the blood vessel or which constitute the blood vessel, okay, they are very tightly arranged. Okay, so they do not allow the extravasation or transport into the other organs. However, the blood capillaries supplying to the this RES tissues. Okay, RES stands for reticuloendothelial system. Okay, so mainly liver and spleen. Okay, so the blood vessels supplying to this uh, liver and spleen, they have large intercellular gaps. These are not tightly arranged. Okay, so these nanoparticle, okay, they can enter into the liver and spleen. Okay, because these large fenestration or large intercellular gaps are present. Okay, the similar physiology is available in the case of cancer or tumor. Tumor also has leaky vasculature. Okay, so these nanoparticles, okay, they have tendency to go either into the RES tissues or into the cancer tumor tissue. Okay, but again, what would be the probability whether they will go inside the tumor or whether they will go into the RES tissues? Okay, so these opsonin protein, okay, these blinking proteins, you can see this green and blue. Okay, so these opsonin proteins, these are present in the blood. Okay, so when you inject these nanoparticles, so this opsonin protein, okay, they immediately attack onto the nanoparticle, and they form some coating onto the nanoparticle. So that is called opsonization, and they forcefully take them to the RES tissues. Okay, so the probability of reaching the nanoparticle at that cancer site or tumor site, it is further diminished or reduced. And these RES tissues, liver and spleen, they are rich in the macrophage. Okay, so macrophages or phagocytes, these are defense cells of our body, and they take up or engulf the particle and they degrade the particle. Okay, so that is the process is known as endocytosis. Okay, so this is the static image of the endocytosis. This particle, okay, so it is internalized by the cell. So initially it forms the vesicle inside the cell, so that is called endosome. Endosome is having like a acidic pH, and it is then fused with the lysosome. So lysosome is a cellular machinery which is equipped with variety of enzyme system which can degrade the nanoparticle. So this degraded nanoparticle, okay, it is initially released in the cytoplasm, and then ultimately it is thrown outside the cell by the process known as exocytosis. Okay, so I will share show one of the like a small movie clip to demonstrate how this endocytosis takes place. And this receptor mediated endocytosis, it is mediated because some receptors are expressed onto the biological cell, and which binds with a particular molecule which is called ligand. Okay, so this ligand receptor interaction, it is key of targeted drug delivery. Okay, so these nanoparticles are selectively taken by the target cell which express those receptors. Okay, so here these LDL receptors, these are present onto the biological cell. Okay, this is just the normal case of endocytosis. Okay, so this LDL particle, low density lipoprotein particle, it is structurally it is similar to the your liposome. Okay, so these LDL receptors are expressed onto the cell surface, and this LDL particle, it is made of some like a phospholipid layer. Okay, so cholesterol molecule are wrapped inside this LDL particle. Phospholipid monolayer is there. Okay, so this structure mimics the liposome or conventional nanoparticle. Okay, this binds with the LDL receptor onto the cell surface. Okay, so this binding of the LDL particle with the LDL receptor leads to this shrinkage of the vesicle cell membrane. Okay, clathrin molecules are recruited, so it ultimately engulfs the particle. Okay, so this is the process is known as endocytosis. Inside the cell, this vesicle uncoats. So these clathrin molecules are detached. Okay, and then uncoated vesicle, okay, because it is having the acidic pH, so pH drops to acidic pH. So this further dissociate the LDL particles, LDL receptor. These LDL receptors are detached, and they are ultimately recycled back and rearranged onto the cell surface. Okay, this is then fused with the lysosome. As I said, lysosome is a cellular machinery which is equipped with variety of enzymes, so which cause degradation of the LDL particle into the cholesterol. Okay, so this cholesterol molecule, okay, it is then initially released in the cytoplasm. Okay, these LDL receptors, these are recycled back or rearranged back onto the cell surface. Okay, for next course of the event. Okay, so these cholesterol molecule degraded particle are initially released in the cytoplasm. Okay, then they are thrown outside the cell by a process exocytosis. Yeah, so this will show the exocytosis process. So this lead to formation of the exosome. Okay, so secretory vesicles are formed. These degraded cholesterol molecule or degraded particle. Okay, they are wrapped in the secretory vesicle that is called exosome. Okay, this vesicle membrane. Okay, this fused with the plasma membrane, and the contents are thrown outside the cell. Okay, so this is a very simple process of exocytosis. Okay, so this actually happens. Okay, so as I said, some receptors are expressed onto the cell surface. Okay, which is responsible for receptor mediated endocytosis. Okay, so this ligand receptor interaction, it is the key of targeted drug delivery. Okay, so every biological cell, it is capable of endocytizing the things, okay, like molecules or like nanoparticle. Okay, and that endocytosis, it is facilitated by ligand receptor interaction, and some receptors, they are overexpressed in a particular disease condition. Okay, so that selectively binds with the particular molecule. Okay, for example, in the case of cancer, this folic acid receptor, these are overexpressed. Okay, so if we functionize the nanoparticle with folic acid, okay, so we can design them as a target specific nanoparticle. Okay, so this is important. We need to have information what type of receptor, okay, they are overexpressed onto our target cell, okay, or cell which we want to target. So accordingly, we can design the nanomedicine. Okay, here. But again, if we can design this ligand anchored multifunctional nanoparticle, then again the major question comes, how to bypass the res uptake. Res means reticuloendothelial system, res uptake. Okay, so if we have this plain nanoparticle or simply ligand anchored nanoparticle, okay, they will simply end up in going into the liver and the spleen, where they will be taken up by the macrophages. Okay, and they will be degraded. Okay, so this res uptake can be bypassed by pegylation. Okay, so peg hydrophilic network of the peg can be arranged onto the nanoparticle surface. Okay, so this lead to repulsion of the opsonine protein. So opsonine protein cannot recognize the exact surface of the nanoparticle. Okay, so this type of like a pegylated nanoparticle or nanocarrier, okay, they are called stealth particle or long circulatory particle. Okay, they are not recognized by the opsonine protein, and they are not recognized by the res system. Okay, so they will remain in the blood circulation for prolonged period of time, and they will have probability of reaching at the tumor site, because tumor also has leaky vasculature. Okay, so the commercial formulation doxil, okay, that is pegylated liposome containing doxorubicin, it is already available in the market based on the same concept. Okay, and further, if we have this targeted ligand present onto the nanoparticle surface, so it will selectively interact and bind with the target cell, and it will be internalized by the receptor mediated endocytosis. But on the normal cell, because we do not have any receptor on its surface, so they will not interact with the normal cell. Okay, so the toxicity towards the normal cell, okay, it can be avoided in this way. Okay, so what I want to convey here, if we have this plain nanoparticle, conventional nanoparticle, okay, so they won't be much effective. They will show rapid clearance from the circulation, and they will show higher uptake by the res organs. Okay, they do not have any target specificity. Okay, so these are useless carrier system, except for the like liver associated disease. These are good for the liver associated disease like leishmaniasis or any other. Okay, but otherwise these are useless system. Okay, to generate target specificity, we can conjugate with the some site directing ligand, but again the res uptake cannot be avoided, so they will show rapid clearance from the circulation, and they again they won't be much useful system. Okay, to bypass the res uptake, okay, so we can generate this long circulating or stealth carrier system by pegylation. Okay, but this pegylated carrier system, although they will have a prolonged circulation half life, but they do not have sufficient target specificity. Okay, so if we want to enjoy the maximum benefit of the targeting, so we should go for the multifunctional approach. Okay, both pegylation as well as ligand anchoring. Okay, so this pegylation will protect them from the res uptake, and ligand anchoring will uh, ensure the selective uptake by the targets. Okay, so this is the basics, means how we can design the targeted nanomedicine. Okay, so based on the same concept, a long time back we developed this methotrexate loaded folic acid conjugate and pegylated this human serum albumin nanoparticle. So albumin is the polymer to make the nanoparticle, and this PEG to make them long circulatory, and folic acid as a targeting ligand. Okay, this is the AFM image of the nanoparticle, and these are the scintigraphic images in the mice. So in one leg, okay, we implanted tumor, and another leg it was free. Okay, it was normal leg. Okay, we injected the formulation through tailweed, and we checked the biodistribution pattern. Okay, so here in the folic acid conjugated our target nanoparticle, highest uptake can be seen at the tumor site as compared to the free drug and this other controlled formulation. And quantitatively, when we did this quantitative biodistribution, okay, so there's a tumor to muscle ratio. Okay, tumor is the target site, this muscle ratio, that is the other leg, normal leg. Okay, so we quantified the concentration. Okay, so this tumor to muscle ratio, target to non-target ratio, it is 20 times higher. Here it is 2, and here it is 40, 20 times higher retention at the tumor site Okay, it was observed using this targeted nanoparticle formulation. Okay, similarly, in the same direction, we prepared this docetaxel loaded, okay, folic acid functionalized, pegylated, this liquid crystalline nanoparticle. Okay, so liquid crystalline nanoparticle, these are also made of some lipid. Okay, but these are different from the liposomal lipid. Okay, so these are also self-assembled nanoparticle. Okay, so depending on the method of preparation, okay, nature of the drug, and the nature of the lipid, so they self-assemble either in the hexagonal shape or cubic shape. In this case, we got this cubic shaped particle. Okay, so we can also call them cubosomes. Okay, these are the particle size and entrapment efficiency data. So we got particle size less than 200 nanometer and more than 80% entrapment efficiency. So that was good. These are the TEM images of the this cubosome, and these according to this schematic, these are the aqueous channels. Okay, so the same can be seen in the high resolution TEM images as well. Okay, so when we tested in the animal model, so breast cancer containing animal, so we divided the tumor bearing animal in two groups. One is a large tumor bearing animal where the tumor size was more than 1000 millimeter cube, and another one, the small tumor bearing animal where the tumor size was less than 1000 millimeter cube. Okay, so we injected the formulation, okay, the marketed formulation Taxotere. Okay, we have also used. So this Taxotere formulation, okay, gave nominal protection. Like in the untreated animal, the tumor growth continuously increased. Okay, so starting from 100%, so up to around 180% increase in the tumor growth was observed. In the case of marketed formulation Taxotere, okay, so slight protection like 20% reduction in the tumor volume and final tumor volume was 80%. Okay, but in the case of our formulation, a folic acid functionalized liquid crystalline nanoparticle, so almost 80% tumor was reduced and 20% tumor was remaining. Okay, this one. And in the case of a small tumor bearing animal, so just after 4 days, okay, so entire tumor, okay, it is cured, and no visible sign of the tumor was observed. Okay, in this case, the Taxotere or marketed formulation gave almost 50% protection. Okay, but our formulation, it gave 100% protection. Okay, another case study I would like to present on this carbon nanotube. Okay, so carbon nanotube, it is also one of the very important carrier system for drug delivery. Okay, so because of the unique needle shaped geometry, it shows good cell internalization by the cell. Okay, it also shows good loading into the onto the nano carrier system, good drug loading, which is otherwise not possible. Okay, and the major drawback with the carbon nanotube, they are not soluble in the aqueous medium. They are very, very hydrophobic in nature. Okay, so if we try to disperse these carbon nanotube, they tend to settle immediately in the aqueous medium. Okay, so whatever the energy input we give, like homogenization, sonication, use of some stabilizer like surfactant, okay, so we could not improve the dispersibility. So this is the problem. And another problem, they are not biodegradable, so they penetrate in any of the vital organs and they remain accumulated for the prolonged period, so they cause toxicity. Okay, but all these problems can be solved or resolved by functionalization of the carbon nanotube. Okay, so for functionalization, okay, so heating with the concentrated acid, it is reported in the literature. So if we do this acid treatment, so it generates hydrophilic carboxylic acid group onto their surface. Okay, so this oxidized carbon nanotube, they are dispersible in the aqueous media, and further we can improve the hydrophilicity by pegylation. Okay, so because these are is carboxylic acid group are there, so we can easily pegylate them. So this further increase their dispersibility and the biocompatibility. Yeah. Okay, so when we started this work, okay, several years back, almost 10 years back. Okay, so we had some confusion this oxidation for how much duration of the time. Okay, because in different reports, different methods were reported. Like some scientists said oxidation or heating with the acid overnight. Some reports said just 2 hours is sufficient. Some said no, it should be minimum 12 hours. Some reports said 24 hours. Okay, so we thought we should optimize the oxidation time. Okay, so we did so many studies. So this pristine carbon nanotube, these are the same images. So pristine carbon nanotube. Okay, so we can see this aggregated bundles of the carbon nanotube. In after 2 hours of oxidation, so this aggregation was somewhat reduced, but still, and the side panel we can see lot of aggregation. Okay, and this is after 6 hours of oxidation. So this individual carbon nanotube can be seen, and this is after 8 hours of oxidation. Okay, so this structural integrity of the carbon nanotube, it was compromised. So these are not having this tubular shape, okay, or cylindrical shape. These are amorphous carbon particle. Okay, we also checked their geometry or morphology under transmission electron microscopy. So this multi-walled carbon nanotube, okay, we could eventually see. Okay, so these different walls can be seen, and the distance between the two walls can also be determined. Okay, using this distance. And the drug doxorubicin, it was physically encapsulated. Okay, so physically encapsulated. So this doxorubicin, it is a fluorescence molecule. So encapsulation of the drug, okay, inside the carbon nanotube lead to reduction in the fluorescence intensity. So this doxorubicin in the free form, it shows some fluorescence spectra. Okay, however, the fluorescence intensity was reduced when the drug was loaded into the carbon nanotube. So maximum reduction or quenching of the fluorescence was observed at this particular ratio 2:1. Okay, means 33% of the doxorubicin it could be loaded. Okay, so this is a very high loading we could achieve, but otherwise in the liposome, so not more than 3% of the drug it is loaded. Okay, but we could achieve 33% loading, so which is very high. And when we tested in the animal model, so these are the self-explanatory images. Okay, so minimum tumor burden, okay, could be seen. Okay, as compared to this free doxorubicin. Okay, doxorubicin loaded in the carbon nanotube, so it shows the reduced tumor volume. Okay, we also checked the toxicity, liver toxicity of this carbon nanotube by measuring the liver function test or this serum biochemical parameters like GOT level, GPT level, MDA level. So in the case of toxicity, the levels they are significantly elevated or increased as compared to the normal value. But in the case of our functionalized carbon nanotube, okay, the levels remain close to the control value. Okay, so that indicated these carbon nanotubes were relatively less toxic. Okay, so in the next we prepared this multifunctional carbon nanotube. Okay, so hyaluronic acid functionalized pegylated carbon nanotube. So hyaluronic acid, it is a good ligand for CD44 receptor, which are overexpressed onto the tumor cell. Okay, so this pristine carbon nanotube, okay, or plain carbon nanotube, they were oxidized, okay, or functionalized. Okay, so this carboxylic acid, okay, group containing carbon nanotube, this carboxylic acid group were further activated using standard thionyl fluoride treatment. Okay, so this was then reacted with this PEG molecules on both sides. Amine group was present. Okay, so this one NH2 group, it is reacted with this acidic group to form this amide linkage, and these are the pegylated carbon nanotube having another terminal amino group on the surface. Okay, so which was reacted with the acidic group of the hyaluronic acid. Okay, so these are the HA functionalized or HA containing carbon nanotube. Okay, doxorubicin was physically encapsulated. So all these reaction, okay, we are monitored or characterized using standard spectroscopic technique like FTIR, thermogravimetric analysis, X-ray photon spectroscopy, and this NMR spectroscopy. Okay, but I'm not sharing all the details. Okay, I will just show some cell culture findings. Okay, so this hyaluronic acid containing carbon nanotube, and these are simply pegylated carbon nanotube. Okay, so both, okay, are taken up by the cancer cell. Okay, but the degree of uptake, it was high in the case of HA containing carbon nanotube. Okay, as I said, this carbon nanotube, okay, they are having the needle shaped geometry, so they can be taken up by the cells, okay, by non-specific mechanism as well. Okay, but the hyaluronic acid containing, okay, they are internalized by receptor mediated endocytosis, because okay, the uptake was completely blocked, okay, when we pre-treated the cell, okay, with free hyaluronic acid, okay, to saturate the receptors. So in this case, the uptake was completely blocked. Okay, so that confirm this, the uptake in this case, it is because of the receptor mediated endocytosis. Okay, this also had some implication in further targeting at the organelle level or cellular level. So inside the cell, okay, there is a cytoplasm, there is a mitochondria, there is a lysosome, there is a nucleus as well. Okay, so we want this doxorubicin. Okay, if we remember the mechanism of action, so doxorubicin acts on the nucleus. So we want, okay, this after internalization by the cell, okay, our uh, this drug or carrier system will go with the nucleus. Okay, so we checked using this differential staining concept. So we stained the cell with a dye known as DAPI. It is a blue color dye, so that selectively stained the nucleus of the cell. Okay, and the doxorubicin, okay, so it is a fluorescence molecule which gives red fluorescence, and additionally we tagged our carbon nanotube with a fluorescence dye, that is Alexafluor 647, AF647, which gives red fluorescence. Okay, so this blue fluorescence is because of the nucleus, and this red fluorescence, it is because of the carbon nanotube. And when we did this overlapping analysis, okay, so we got perfect overlapping of this blue and red fluorescence. It means the carbon nanotube and the nucleus, they are, or they were at the same place, but the same synergy was not observed in the case of pegylated carbon nanotube. Okay, so although they are taken up by the cell by a non-specific pathway, okay, but they cannot target uh, further to the nucleus, which is desirable organelle. Okay, then we check, okay, all other tests to induce the capability of apoptosis. Okay, so using different assays like an exin B assay, tunnel assay, rubidium iodide or diphenylamine assay. Okay, so this free drug, okay, it was able to induce the apoptosis. Okay, so these are some values like 60, 65, 50. Okay, but the same drug, when loaded into the carbon nanotube, so it induced higher degree of apoptosis. Okay, like 75, 78, so which is higher. Okay, so we can say the efficacy of the drug, it was significantly increased. Okay, so moving forward, so I will show some lights on the co-administration of the two drugs for the synergistic anti-cancer therapy. Okay, so present this combination therapy, okay, with two or more drug, it has emerged as a very promising strategy to suppress the cancer drug resistance, and we can get synergistic anti-cancer response. Okay, because the two drug, okay, they act on the different stages of the cell growth cycle. Okay, so otherwise what happens, if the drug acts on the like G phase, so the cells which are present in the S phase, okay, so they will not be killed. Okay, so that ultimately lead to development of the drug resistance if we treat the cancer with just one drug. Okay, monotherapy. So that's why if we are using combination therapy, so some drugs which are present in the G phase, so they will be killed by a one drug. Some drugs, okay, some other cells which are in the S phase, they will killed by the other drug. Okay, so this is the advantage, we get the synergistic anti-cancer response as well as we can overcome the drug resistance. Okay, but there are some limitation with the conventional chemo combination therapy. Okay, because the two drug, they have a different solubility profile, different permeability profile, so they will behave differently inside the body, like their ADME profiling, okay, or their biodistribution would be different. Okay, but for the synergistic efficacy, we need, okay, their availability at the cancer site at the same time and the same rate. Okay, so that is the problem. Okay, so here we designed the combinatorial bioconjugate of the two drugs. Okay, so because the two drug molecule would be present, okay, in the same molecule, okay, two drugs would be present in the same molecule, so we can get good solubility balance or solubility profile, synergistic response. It can overcome the like resistance problem, and because the two molecules or two drug would be in the same molecule, so we can have the like a similar or uniform pharmacokinetic and cell uptake, means the availability at the cancer site, it would be at the same rate. Okay, so how it can be designed? So two anti-cancer drug like gemcitabine, it is a anti-cancer drug one. Okay, it is a highly hydrophilic or water soluble in nature, and another anti-cancer drug like docetaxel or curcumin, which are practically insoluble in water. They can be conjugated together with the help of this PEG, polyethylene glycol, as linker. Okay, so we can get good solubility. Okay, prolonged circulation because of this PEG and the resistance to metabolic degradation. Okay, so ultimately we get this improved pharmacokinetic and improved anti-cancer efficacy or synergistic anti-cancer efficacy. Okay, this is ultimately what we are going to achieve. Okay, so we selected this gemcitabine and the docetaxel as a combination. Okay, so because these two drugs, they act on different target. Okay, so different mechanism of action. Okay, so we can get synergistic anti-cancer efficacy, and importantly, they show non-overlapping toxicity. This is also important. Okay, like if the docetaxel cause a liver toxicity, then this gemcitabine, it will cause hemolytic toxicity. Okay, otherwise if the toxicity is same, then it would lead to a very high or serious toxicity, and this is FDA approved combination. Okay, of course in the conventional form. Okay, but the problem with the individual therapy, okay, or conventional combination therapy, this gemcitabine, it is a highly water soluble drug and it is highly unstable drug. Okay, so its half life is just less than 90 minutes. Okay, rapidly metabolized into the inactive metabolite, that is called DFDU. Okay, that is deoxydifluorouridine. Otherwise it is a cytidine analog. Okay, and on the other hand, this docetaxel, it is practically insoluble, and even the marketed formulation, it contains twin 80 as organic like a surfactant, and this alcohol as a organic solvent. Okay, which is not at all recommended for the parenteral or intravenous administration. Okay, but as a special case, this formulation, this taxotere, it is approved. Okay, this containing organic solvent and surfactant. Okay, and other problem is, okay, they are having different or opposite solubility profile. So one drug may go at in one direction, other drug will go in other direction. So we cannot ensure their delivery at the tumor site at the same rate, okay, or same time. Okay, so what with this gemcitabine and docetaxel, okay, they were linked together with the help of linker. Okay, so different type of linker we have optimized, like direct conjugation, so that is zero length linker, or short chain linker, so that is just one amino acid glycine or lysine to amino acid, or long chain linker, that is different grades of the PG. Okay, so we prepared this amphiphilic dual drug conjugate. Okay, so because both hydrophilic and lipophilic groups are present in the same molecule. Okay, so this behaves like a amphiphile. Okay, so hydrophilic group, it is because of this gemcitabine and the PG, and the lipophilic group, it is because of the docetaxel. Okay, but ultimately this molecule becomes amphiphilic in nature or amphiphile. Okay, so that showed the self-assembling characteristic. Okay, so we could prepare this self-assembled nanoparticle containing both docetaxel and the gemcitabine. Okay, we also determined the critical micelle concentration of this amphiphile. So it was around 7.83. Okay, so if we disperse this molecule bioconjugate above this concentration in the water, concentration above this concentration, so it can result in self-assembled nanoparticle. This is the AFM image, this is the SAM, and this is the TEM image of the self-assembled nanoparticle, and the size was around 124 nanometer. That was very less. In vitro release we perform, okay, so at pH 5.5, that is the tumor interstitium pH, which is slightly acidic, so they show higher release as compared to at pH 7.4 in physiological pH, which is desirable. Okay, as I said, this gemcitabine, it undergoes rapid metabolism to the inactive metabolite, that is DFDU. So that was also checked. So in the case of free gemcitabine, okay, 62% of the DFDU it was generated. Okay, but in the case of the self-assembled nanoparticle, okay, so only 14% of the DFDU it was generated, and remaining in the drug remain intact, okay, or undegraded, or it is not degraded. Okay, we also checked the cellular uptake. So higher cellular uptake of the self-assembled nanoparticle can be seen as compared to the this individual drug or free drug. Okay, we also determine the cell internalization pathway. Okay, so of course, so many studies we have done. Okay, using so many inhibitor or different transporters or different inhibitors for different transport pathway. Okay, so we concluded this self-assembled nanoparticle, they are internalized by the receptor mediated endocytosis process. Okay, we also checked their capability to induce apoptosis in two different cancer cell line, MCF7 and MDMB. So both are breast cancer cell line. Okay, so these green cells, these are a healthy or live cell. This red cell, these are the apoptotic cell or dead cell. Okay, so the ratio of this uh, red to green fluorescence, it gives apoptotic index. Okay, so higher is the apoptotic index, it means higher is the efficacy of the drug. Okay, so in the case of free gemcitabine, it is around 0.42. In the case of free docetaxel, it is around 0.54. In the physical mixture or conventional combination therapy, it is 0.75. But in the case of self-assembled nanoparticle, it is close to 1.98. So it again shows the highest efficacy. Okay, the similar trend was observed in other cell line. Okay, we did the in vivo pharmacokinetic also. Okay, so the half-life and the plasma concentration of both gemcitabine and the docetaxel using self-assembled nanoparticle, it was significantly increased by 5 times and 4.8 times, okay, as compared to the individual free drug. And we also checked the qualitative biodistribution using this fluorescence microscopy. So we got a very high tumor drug concentration. Okay, this is the tumor, and in other body organs or like a heart, liver, kidney, the concentration was negligible or it is very less. Okay, however, in the case of free drug or free dye, so the uptake at the tumor site it was minimum, but uh, uptake in the other organs was predominant. Okay, we checked the anti-cancer efficacy as well. Okay, so we got minimum tumor burden using our self-assembled nanoparticle. Okay, so approximately 40% tumor was just remaining, less than 40%. Okay, and we also checked the toxicity. So organ toxicity like liver toxicity by measuring this serum biochemical parameters like EST level, ALT level, MDA level, and the nephrotoxicity like this blood urea nitrogen level and the serum creatinine level. In the case of toxicity, I mentioned earlier, the levels elevated or increased significantly as compared to this normal or healthy values. Okay, but in the case of our nanoparticle, self-assembled nanoparticle, the levels remain close to the healthy value. So it means or indicated that our nanoparticle, they were less toxic. Okay, we checked the histopathology of the major organs, okay, to determine the toxicity. Okay, so liver, spleen, and the kidney, and then this hemolytic toxicity, damage of the red blood cells. Okay, so this is a same image of the RBC. So this shows like healthy RBCs. Okay, this is after treatment with this free gemcitabine. Okay, so we can see that distorted shape crenation is there, star shaped RBC. Okay, so that is not desirable. But in the case of this self assembled nanoparticle, the morphology looks like healthy RBCs. Okay, so next case study, or moving ahead on to the another topic, nanomedicine for the oral cancer therapy. Okay, so in the case of intravenous delivery, okay, we need to visit hospital, okay, to take the injection. So that is expensive also, and it is not patient friendly. Okay, so it would be good that if we can have medicines which we can take through oral route. Okay, so oral route is more easiest or convenient way of drug administration, but unfortunately many drugs, they do not absorb, okay, significantly after oral administration. Okay, so what is the problem, why the drugs are not absorbed, and how this nanotechnology can improve the absorption of the drug? Okay, so I will take just 2 minutes to explain this concept. Okay, so there are two different type of cells present in our small intestine. Okay, so one is called enterocytes and another is called M cell. So the normal drug molecule, they are absorbed via like passive diffusion via this enterocytes. So for that, the first step is the solution or dissolution of the drug. Okay, so if the drug is not soluble in the aqueous medium, then dissolution would be poor and drug will not be absorbed. Okay, besides dissolution or solubility, the permeability of the drug, it is also important. So if the drug is not permeable or drug is having a low lipid solubility, then it will not be able to absorb or permeate across this intestine. Okay, another problem is if the drug is unstable in the GI condition. Okay, like many drugs, they can, and they are degraded at the acidic pH of the stomach, or enzymes present in the is intestine or in the stomach, so they cause degradation of the drug. Okay, so that is again the problem. And another problem is peak glycoprotein efflux system. So PGP efflux system, okay, so which act as a efflux transporter, it is expressed onto the intestinal cell membrane. So many drugs, they are substrate for this PGP efflux system, so they are not absorbed. Okay, another problem, if the drug molecule can overcome all these barriers, but still, okay, the major fraction, okay, during the absorption process, major fraction is metabolized in the liver. Okay, that is called first pass metabolism. Okay, so all these challenges can be overcome if we uh, develop the formulation in some nanocarrier system. Okay, so encapsulation of the drug in the nanoparticle, so it offers protection from the premature degradation like acidic pH or digestive enzymes. Okay, and from this nanoparticle, they are directly taken up by the M cells, and from M cells they are transported into the lymphatic system. Okay, so they are taken up by the process endocytosis. So for the endocytosis, there is no need for solubility, there is no need for the permeability. It is direct particle uptake by these M cells. Okay, and this PGP also does not express onto this M cell, and from M cell, the nanoparticle or drug molecule, they are absorbed into the lymphatic system. Okay, so it also bypass the first pass metabolism in the liver. Okay, this can be explained by this schematic. So when free drug is administered, okay, some problem with the formatting. So when free drug is administered, okay, so either it will not absorb, but if it is absorbed, then it enters into the liver. Okay, where major fraction can be metabolized. But from the nanoparticle, okay, they are absorbed into the lymphatic system. Okay, so from lymph they directly enter into the liver. Okay, so it bypass the first pass metabolism in the liver. Okay, besides it, there can be other n number of mechanisms which can improve the oral bioavailability. Like if we have nanocrystal, so it increase the surface area, so it increase the dissolution velocity. Okay, so for BCS class 2 drugs, so it increase the overall absorption. Okay, chitosan nanoparticle, okay, so they are reported to open the tight junction. Okay, and some other mechanisms are also there. Okay, so there can be n number of mechanisms. Okay, so tamoxifen, it is a well-known anti-cancer drug for treatment of breast cancer. Okay, the problem with tamoxifen, okay, so it is a BCS class 4 drug, poor solubility, poor permeability. Okay, to improve the solubility, tamoxifen citrate, it is commercially available. Okay, but this citrate salt also undergo precipitation in the acidic media of the stomach. Okay, and it also undergo extensive first pass metabolism. Okay, and it is a substrate for P-glycoprotein efflux system. So the absolute bioavailability for tamoxifen citrate, it is just a 15% or 20% maximum. Okay, because of these reasons. Okay, and every time we are taking tamoxifen and it is metabolizing in the liver, so it lead to oxidative stress onto the liver and it cause hepatotoxicity upon chronic administration. Okay, so we thought of developing the formulation using quercetin. Okay, so there can be n number of approaches to improve the oral bioavailability, but we selected this self-emulsifying drug delivery system. Okay, because this is nothing but, okay, this is a isotropic clear isotropic clear oily liquid. Okay, so mixture of oil, surfactant, and co-surfactant in which the drug is solubilized, and this oily mixture can be filled in the soft gelatin capsule shell. Okay, or it can be absorbed onto some carrier system, and we can compress the tablet. So when this formulation is taken orally, so this form nanoemulsion inside the body without the need of any external agitation or homogenization. So this is a very simple technique, and no level, no sophisticated technology or instrumentation or equipment it is required. So it demonstrate high level of the industrial scalability. Okay, so we prepared the formulation, and the quercetin we have added quercetin as an antioxidant. So our motive was, okay, so to reduce the oxidative stress which cause hepatotoxicity. So our main objective of adding quercetin was to reduce the hepatotoxicity of the tamoxifen. Okay, so we prepared the formulation, we did lot of optimization, and we prepared the formulation, and we checked the bioavailability. So we found, okay, without quercetin, okay, using simple snets, okay, the formulation increased the bioavailability by 3.89 or approximately 4 times, fourfold as compared to tamoxifen, marketed tamoxifen citrate. But with quercetin, okay, the bioavailability was further increased up to 8-fold. Okay, so our objective of quercetin addition was to reduce the hepatotoxicity, but we got further improvement in the bioavailability. So when we cross-checked the result, and we did some analysis or reason finding, so we noticed or we observed that quercetin is well-known inhibitor for the P-glycoprotein efflux system. So that was the reason it is increasing the bioavailability of tamoxifen. And we checked the anti-cancer efficacy. So this was the, like, minimum tumor burden was observed with tamoxifen quercetin sets as compared to this marketed formulation. Okay, so it was most effective, and we checked the hepatotoxicity. So we did not find any significant hepatotoxicity like MDA level or AST level, ALT level, GSH level. So a lot of serum biochemical parameters we investigated. The last case study I would like to present on the localized transdermal delivery for breast cancer therapy. Okay, so far we are taking the drugs either through intravenous route or through oral route. Okay, so ultimately the drug reaches into the systemic circulation, and from systemic circulation or from the blood, it reaches at the cancer site. Okay, so like a breast cancer, so unnecessary the other body tissues, they are exposed with the drug. Okay, so this is the problem. So we thought, can we develop some formulation in the topical cream or gel base, okay, so that when we apply onto the breast cancer region, so it will be effective locally, and so we can avoid our other like blood or other body tissues to be exposed from the drug. Okay, but again the topical delivery okay, transdermal delivery of the drug, it is a challenging task. Okay, so we prepared this high permeation vesicles. Okay, so different permeation enhancer. Okay, we did lot of optimization, almost 6 or 4 different permutation enhancer. Okay, in permutation combination we have identified, and we selected. Okay, so that scope, that is synergistic combination of the permutation enhancer. Okay, so initially using some fluorescence dye, we checked the skin permeation or flux of the molecule. So we got 12-fold enhancement in the case of high permeation vesicle. Okay, as compared to this nanoemulsion, liposome, transfersome. Transfersome is a well-known carrier system for transdermal delivery. Okay, so here it is also clear or self-evident from this fluorescence images. Okay, HPV, okay, it is highest amount is going inside the skin. Okay, so lot of optimization we did, and this is the skin permeability profile. So it shows highest permeation across the skin, HPV based formulation. Okay, however, this free drug, free docetaxel, it was not able to cross at all the skin barrier. Okay, so this was the problem. Okay, and we compared the findings with his co-mixture, means just a permission enhancer and liposome as well, and we got encouraging results with the uh, high permission vesicle formulation. The in the cell uptake also in the cancer cell, so highest uptake can be seen in the case of HPV based formulation. In the cell cytotoxicity, that is the in vitro anti-cancer efficacy, so HPV based formulation showed minimum IC50 value or highest cytotoxicity. Apoptosis also we checked, so we got highest apoptotic index in the case of HPV formulation. Okay, and finally, when we checked in the in vivo anti-cancer efficacy cancer model, so minimum tumor burden using our HPV based formulation was observed, and we compared the efficacy with the intravenous formulation, marketed formulation of docetaxel. So we got almost equivalent efficacy. Okay, using our HPV, means topical formulation. So that was the good thing, and we also checked the qualitative biodistribution or uptake inside the tumor. So we got highest uptake inside the tumor using this HPV based formulation. Okay, we did the toxicity analysis also, so we got the minimum toxicity. So I will not focus on this result because these are the repeated findings. Okay, so one may complain if this formulation contains lot of surfactants, okay, or permeation enhancer, so it may be irritant to the skin. So that aspect was also checked. Okay, so skin histology. Okay, so we got like a minimum damage to the skin. Okay, so the morphology looks like the healthy skin. The same was also revealed using transepidermal water loss values. Okay, so our formulation was non-irritant. Okay, so it was a good formulation. So I would stop here. These are the key research areas of my lab. Okay, multifunctional nanocarriers for targeted drug delivery, gene delivery via non-viral vectors, non-invasive vaccine delivery, oral bioavailability enhancement, topical and transdermal drug delivery, co-delivery of anti-cancer drugs for synergistic efficacy, and lipid products for effective drug delivery. Okay, so some of the aspect I have covered during this presentation, and these two books we have published, one on targeted drug delivery by Springer, and another on this targeted nanomedicine for the cancer nanomedicine by Elsewhere. Okay, and this is the most prestigious moment of my life, and I received this Young Scientist Award from Dr. Kalam, former president. And thank you very much for your attention. And if you have any question, then I will be happy to answer. Thank you.
Thank you, sir, for enlightening us on the design of targeted nanomedicine for the cancer therapy. Now the session is open for questions.
Good evening, sir.
Yes, good evening.
Myself, Sumant, from Vishnu College of Pharmacy.
Yes.
Doing pharmacy. Sir, I have one doubt
Yes.
about the silver nanoparticles. I'm doing the project silver nanoparticles, sir. I hear
Okay.
the words like metal nanoparticles was toxic. Is it real, sir?
Yes.
Is it useful to doing, sir?
They are good for like just for diagnostic purpose. They are okay, but for the therapeutic purpose, they are not good. Unless
Sir,
otherwise, if you can make them highly specific for the target, okay, then they may be useful, but otherwise no, because heavy metal poisoning would be there.
Sir, what was the better size range about the nanoparticles, sir? Good size range.
Best size range is less than or approximately 100 nanometer.
Thank you, sir.
Yeah. Yes. So any other question?
No question. Dad?
Sir,
Yeah.
sir, this is P. Sai Prolika studying M-Pharmacy second year, sir. So Department of Pharmaceutics. And I am formulating a mucoadhesive nanoparticle, sir, for treating dry eye problem. Which polymer is suitable for the formulations?
Yeah. So there are n number of mucoadhesive polymers, like Galan gum is also reported. Okay, so you can use any one. Okay, but you just need to check whether means it should not cause any irritation, ocular irritation. It should not cause.
Okay.
Yeah.
Okay.
So
Thank you.
that is important. Otherwise, any polymer is good.
Okay. Thank you.
Yeah, and some polymers they shows mucoadhesiveness that is pH dependent. So that aspect also you need to consider, because the ocular pH or the pH of the ocular cavity, it is close to physiological pH 7.4. Okay. So they should be mucoadhesive at that particular pH. Like a chitosin, it is reported it is mucoadhesive, but at this means it is mucoadhesive at acidic pH only. Okay, it loses its mucoadhesiveness when it comes to pH 7.4.
Yes, sir.
Yeah.
Okay, thank you.
Yeah.
Now, sir, as a token of gratitude from Sri Vishnu College of Pharmacy, kindly accept the certificate, sir. The certificate of appreciation will be sent through the mail, sir.
Yeah. Thank you.
Now, my friend Durga will address the vote of thanks.
Thank you, sir. Thank you, sir. James,
Thank
sir.
you. Yeah.
Myself, Durga Satyavati from 2nd M.Pharm, Department of Pharmaceutics, Sri Vishnu College of Pharmacy. I thank our guest speaker, Dr. Sanyog Jain, for throwing light on design on targeted nanomedicine for cancer therapy. Your thoughts have enlightened our minds. And thank you, sir, for adorning the occasion and sharing your opinions today. I cordially thank each and everyone for making this session more interesting with your presence. So please accept our certificate. We will send it to your mail. And I also thankful for the guest speakers of first and second day session, Dr. Vandana Patravali ma'am and Dr. Via Sinha sir, for making this event more successful, sir. Thank you, sir.
Yeah, thank you.
Sir, with your permission, we are signing out, sir.
Yeah, sure. Thank you. Thank you.
For all the participants, the feedback forms will be sent to the group. So please kindly fill the feedback forms by today evening, and the respective certificates will be sent to a mail within 3 days. Feel free to ask any questions and contact us if there are any questions. Thank you all for the participation and making this webinar a grand success. Thank you all.